The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, welcome to episode 99, yes, nearly at the century of the eLife podcast. I'm Chris Smith from The Naked Scientists, and in this episode, why a group of monkeys have taken to washing their food, what does learning multiple languages do to our brains, and the cancers that manipulate their hosts. We'll find out how. But first, a university compost heap has turned up Finland's first documented giant virus. Gabriel Almeida, now at the Arctic University of Norway, had been collecting samples from all kinds of environments to try to find examples of one of these agents. These weird entities were first recognised for what they are, massive viruses bigger than bacteria in many cases, only about 20 years ago. Previously, scientists thought there was some strange form of bacterium, but they've since been detected in many different environments. They often have massive genomes, the workings of which we haven't yet unpicked, but which might surrender some useful secrets, including the recipes for novel antibiotics. And scientists think they might be evolutionarily extremely ancient, perhaps pointing us towards how early life might have operated. Well, now Finland can say it's joined the club, and it has one too.
This paper shows the isolation of the first giant virus in Finland. So a giant virus is a virus which is larger than conventional viruses. What makes them giant are two things. One is their size itself, and they also have larger genomes. So it means that they codes for more genes than most common viruses. And that makes them produce a lot of interesting bioproducts.
Where are they generally found? Or in other words, when we've looked so far, where have we found them?
The original one was found from an hospital sample in Bradford in the UK. But after that, we know that they are spread in nature. They are very common to be found in marine samples, in soil, and many other places, even in clinical samples. And this one we found, for example, came from composting soil sample. So it means that they are really widespread.
It came from a compost heap?
Yeah.
And so did someone actually go, or did you go looking in a compost heap to find it? Or were you looking for something else and found this?
I was building a collection of samples in Finland. So I was collecting everything I could, from lake water, from soil, from aquaculture-related samples. And we had this composting place in the university, and I collected a small amount of that. And then I tested, and it was positive for the virus.
How did you actually test it? In other words, when you were just given this soil sample, how did you then flush the virus out?
For the giant viruses, they're special in a way, because for most of them, they need to be eaten. So that's why we often use amoeba to isolate them, because the amoeba is actively taking food sources from the environment, and when they eat one of these viruses, they get infected. So the process is basically to take the sample, make it in the liquid form, and then mix that with a growing amoeba culture, and the amoeba are going to eat everything that's in there. And if they find a virus, then they get sick, they die, and you can see that by the microscope that they are dying, and we know that likely a virus is there.
And when did it become apparent to you that you'd got one of these viruses? Was that because your amoebic culture became unwell and you reasoned, well, something's killing it, it could be a virus?
Yeah, exactly. So from 24 to 48 hours later, plates where this sample was added were completely clear of amoeba. Everything died. And we just take a small amount of that to infect more amoeba, and we confirm that it's killing the new batch of amoeba. And then it's not too difficult to grow enough of the virus to extract DNA, the genomes, and sequence that. And once you know the DNA sequence, you can be sure that it's a virus and also which virus it is.
Was it the genome that gave you the clue that you got a new one and that it was Finland's first?
Yeah, exactly. So when we got the full genome sequence, we could see that it was a virus. Also discovered it was related to other known giant viruses. Then we were able to check the genes in details and see their peculiarities and be sure that we had a new one in Finland.
Did you image it? So have you only interrogated it genetically, or have you actually been able to see it now?
So I actually did a lot of imaging as well, based on electron microscopy. And a big part of this paper was actually the cryo-electron microscopy imaging. And after taking images of thousands of these viral particles, reconstruct this structure in very small details to determine how the virus is built. And that's important for knowing how it stays stable in the environment and also how the proteins interact to kind of protect the DNA and make it viable for a while.
What does it look like and how big is it?
So it has around 200 nanometers. Among the giant viruses, a small one. We have some which are 5 times larger than that.
Yeah, indeed. I mean, that would be about twice the size of a flu virus, wouldn't it? Or twice the size of a coronavirus that causes COVID-19. It would be about twice as big as that.
Exactly.
And in terms of its genetic code, how big is it?
So it has 360,000 bases. In comparison, it's going to be like 10 times larger than a phage, for example, much times more larger than corona, like 10 times larger than corona as well. And out of all these bases, it codes for almost 400 genes.
Well, that's quite a lot, isn't it? I mean, because if we take, say, chickenpox or the herpes virus that gives you cold sores, that's got a quarter of that number of genes. That's a lot.
Exactly. And what I find interesting is that it contains genes only found on this virus. So 3 out of these 388 genes are only found on this isolate, and we don't know what they do. So we can have some surprises there. And anyway, 67% of these genes, they are hypothetical. So we know they're code for something, but we don't know they function. So there's a huge biotechnological potential here that you could explore.
Why do they have such massive genomes? Do we actually see all of these genes working? Are they genes that are absolutely critical to the virus life cycle?
So no, they have a lot of accessory genes, and as mentioned, we don't know about most of them. But it has been shown in the literature that some can bring new traits to the host. Some might have hints of metabolic pathways that they also may make the host express. And we have some theories here that they also use these genes to compete with other organisms when infecting the amoeba.
And have you watched them replicating? Can you see where inside the amoebae they grow? Do they go into the genetic material like a herpes virus, or do they grow in the cytoplasm, a bit like a sort of monkeypox virus or a smallpox virus?
Yeah, we were able to see that. So they get taken in by the amoeba, and their replications are in the cytoplasm. They don't get into the nucleus. And they tend to form viral factories. So they are these big electron-dense masses inside the cell where the particles are produced. And for these virus species in particular, they also tend to form some vesicles containing several viruses inside the cell. And that's important because when the cell dies, these vesicles, they form like these clusters of viruses on the environment. And it makes it easier for the new amoeba to find and eat them to be infected.
It's a fascinating phenomenon, though, isn't it? Thinking big, why do you think these things even exist? And where do we think they came from in the first place? How did they evolve? Because they don't seem to bear much resemblance to much else.
They are likely very ancient. When you check the DNA sequences from the environment, you find them everywhere, including on the deep sea vents, for example, where life likely originated. So at least some of the branches of this group of viruses might be as old as life itself.
Is there anything we could use them for, apart from the fact they're academically fascinating? And there may well be some unusual biology locked up in those genes that we can slowly uncover. Do they have any applications that we could exploit?
Yeah, for sure. We have lines of work here in Norway now, trying to untap these hypothetical genes they have and see how they can be used for biotechnology. And one point of investigation we have is whether some of the genes are being used by the virus to kill bacteria. And if that's the case, then we might have something like a viral antibiotic that could be applied in clinics.
An amazing story. Fascinating what you can find in a compost heap, isn't it? Gabriel Almeida there. Monkeys washing their food has provided scientists with an opportunity to understand how animals weigh up the cost of a clean meal against their place in the social pecking order. Taking time out to rinse sandy food ultimately limits how much food you can end up eating, but forego the wash in favour of a greater energy intake to preserve your place in the social hierarchy, and you also end up with sand-damaged teeth that might shorten your life. It's a price that some top dogs are clearly willing to pay for the reproductive opportunities that it opens up. Nathaniel Dominy, who did the work, is at Dartmouth College.
Shortly after the tsunami of December 2004, Researchers were moving on boats up and down the coast of Thailand and Burma doing biodiversity damage assessments. And they were really caught by surprise when they noticed that there were some monkeys on the beach using stone tools. And this is in Thailand. Primarily, they were using them to harvest shellfish, oysters, mussels, that sort of thing. These primates eventually became quite famous, and so tourists started to come to observe these tool-using monkeys. As tourists are wont to do, they wanted to get a closer look at the monkeys, so they started throwing some fruits onto the beach, and this would happen once a day. And the monkeys got used to this. They could hear the boats coming, and they started to expect food provisioned by these tourists. The problem, though, is that the food lands on the sand and it becomes very sandy. And so the monkeys quickly figured out that they could take the food over to the ocean, dunk it, and remove the sand. That was pretty exciting because this recalls a very famous moment in primatology where something similar happened in Japan. And so those instantaneous events of food washing inspired us to conduct an experiment where we gave them foods to elicit food washing to see if it would happen.
What might be the reason why an animal wants to wash food? I know why I would wash the food, because sand tastes awful and the sensation on your teeth is horrible. Is it likely that the monkeys are thinking along the same lines?
It stands to reason, right? It's a relatable problem. Anyone who's thought about having a picnic at the beach, right? It sounds like a good idea in theory, but in practice, yeah, that sand in your mouth, it's so objectionable. And so in 1952, when monkeys were first observed washing sweet potatoes that researchers had given them, the presumption was that they felt the same way, that they didn't like the feeling of sand in their mouth. And so that was the initial motivation to wash their foods. And so that idea that it was a deliberate attempt to eliminate irritable substances on the surface of their food has prevailed ever since. But no one has ever actually bothered to test it.
And how did you test it?
So we had 3 trays and we gave them cucumbers, and that sticky surface of the cucumber is really good for adhering sand. And we gave them 3 options. We gave them a really sandy option, a non-sandy option, and something in between. If the monkeys care about sand, the amount of time they spend cleaning it should be proportional to the amount of sand on the food. And that's exactly what we found. So if a cucumber slice was exceptionally rich in sand, they spent a lot more time trying to remove it.
Thing is that whenever there's an action, there is a consequence. And one of the consequences with this is it's going to incur a delay before they get their reward. And depending upon how sandy it is and how far away the water is, there's an energy cost, isn't there? So what are the impacts of that? Is there a trade-off? Do they kind of say, well, I know how far I'm going to have to take this to get it washed, and therefore I'll tolerate a little bit of sand because the cost will be lower to me in terms of the disadvantage of walking all the way to the water? Or did they always wash it regardless?
You raise a really great point. And that's what we found is that some animals were making shrewd calculations. It just wasn't worth the time to walk to the ocean. And that's how they do it. Because the cucumbers are in their hands, they have to rise onto their hind feet and they move bipedally, a little bit like a human does. Their muscles and bones are not well suited to walking on 2 feet. And so they incur a very high energetic cost. And then there's an opportunity cost. If your hands are filled with cucumber slices and you spend time going to the water to wash them, then there's other cucumber slices that you're not eating. And so we found that some animals would tolerate a little bit of sand on their food because they could eat the cucumber slices more quickly, whereas other animals were strongly averse to the sand, and they would pay that cost, and they were willing to go to the water to wash it more effectively. And interestingly, those decisions seem to vary as a result of their rank and the social hierarchy. So really dominant animals, they have really high energetic needs. A dominant animal needs to fight off other animals. It needs to lunge at them to scare them away. It's constantly on guard. And those behaviors carry very high costs. And so they have to eat a lot and they have to eat quickly to pay for those high energy expenditures. And so we found that it was the dominant animals that just couldn't be bothered, that walking to the water to clean their foods properly was just too costly for them. It would take too much time. And so they actually just brushed it with their hands just a few times and just put it in their mouth. And we could calculate that a lot of sand was going into their mouth. And that was probably ruining their teeth. Sand is very damaging to your teeth. It can cause chips. It can certainly cause a lot of wear. And so we think they were paying a long-term price, that they were shortening their lives probably by refusing to wash their foods properly.
How does this align with our theories of how hierarchy affects these sorts of behaviours? It sounds like that does gel with some of the evidence we already have. And also how we make trade-offs through life in terms of things that we do that we know are health advantageous or health costly, but they're reproductively ultimately advantageous as well. Because that's what this comes down to, isn't it?
That's right. There's this idea in evolutionary biology, and it's called the disposable soma hypothesis. And basically, what it means is that some of your tissues in your body are dispensable, and teeth might be one of those things. There's a very famous study from the UK focusing on red deer, and male teeth wear quickly. So it seems like the strategy is you need energy right away. You want to find mates as quickly as possible. And the importance of doing that far outweighs the benefits of prolonging your life. That's the price you're willing to pay because you get more mating opportunities and you get more food quickly earlier in life. And so we think these monkeys were making a shrewd calculation and just understanding that the sand was bad for their teeth, but they were willing to pay that price because they were getting more mating opportunities in the short run. It's a live fast, die young kind of strategy.
Nathaniel Dominy from Dartmouth College there. This is the eLife podcast from The Naked Scientists, and I'm Chris Smith. In a moment, how tighter control of glucose can keep neonatal sepsis at bay, and the tumour that makes its host grow extra tentacles. But why? Before that, though, people with more grey matter in the auditory areas of the brain should be better linguists, right? Wrong. It turns out, based on findings from a new study, that the more languages a person speaks, the thinner the cortical tissue in the auditory brain becomes. Specifically, it's one region, the transverse temporal gyrus, where greater lifetime language exposure and a greater divergence of those languages, in other words, the more different they are, the greater the effect where this is seen. Study author Olga Kopinska from the University of Vienna thinks it's because a thinner cortex is a more efficient structure honed by language experience. But were the subjects born that way, or did their lifetime linguistic experience mould their brains like that?
Bilingualism shapes the brain, but in our research, we're going beyond bilingualism and we look at people who speak anywhere between one language to 7 languages. So the subjects were healthy individuals who underwent MRI scanning a couple of years ago at University College London in a large-scale study investigating relationships between language processing, language experience, and we looked in particular at the structural scans, so scans where we can distinguish differences in how the brain is shaped.
So because you know which bits of the brain previously have been linked to language, you're saying, let's look in this group of people at this region of the brain in detail and see if there are any structural changes that map onto their linguistic abilities.
Exactly. And not only particularly linguistic abilities, but experience that is linked to the sounds of different languages. So we know that the brain processes sound in structures that are termed the auditory cortex. And we know that the structure is very, very diverse across individuals. My auditory cortex will look very different from your auditory cortex and other people's. And we wanted to zoom in at this structure to see if this variable structure has any neuroanatomical links with people's lifelong language experience.
What did you see when you did this? Was there a relationship between people speaking more languages and more variation in this sound decoding bit of the brain?
Yes, this was very exciting. In this group of approximately 200 people, we saw that there is a lot of variability in how the auditory cortex is shaped. So some people have one bit of cortex devoted to sound processing, others have 2 or 3. And when we looked in detail at the structure of these bits of cortex, we isolated the second transverse temporal gyrus as being specifically related to someone's language experience.
Therefore, if you were to almost draw a graph between how many languages someone speaks and the size or shape of that bit of brain. What's the relationship? What's the pattern there?
The more languages you speak and the more diverse those languages are, the thinner this brain structure is. And it means, in the context of this study, that people who have more experience in different languages need less tissue in the auditory cortex to do its job, basically.
Because initially that seems a bit paradoxical, doesn't it? Because we all talk about the grey matter being the processing power of our brain. And then you say, the more languages I speak, the less of it I've got. But you're arguing this is because it's working more efficiently. It's been presumably honed better by language experience, which means the processing there is about as good as it's going to get, which is why you have less of it.
That's what we think. And when I think about these results, I often have the image of marathon runners who, are extremely efficient and are very, very thin. They have a lot of muscles, but those muscles are shaped in such a way that they're able to run for a very long time.
Do you think, though, that it is that people who have that part of the brain being thinner are better at language? Or did it become thinner because they got better and trained in language? I'm thinking of the studies that looked at the hippocampi, for instance, of taxi drivers, where there was a claim that learning the streets of London did change the shape of one part of the brain, the hippocampus, and you could demonstrate that this occurred with the learning. Is this change you're seeing downstream of the learning, or was it there to start with in very accomplished linguists?
That's an excellent question, and it's actually difficult to draw this conclusion from our study because we look at only one time point, but we have some reason to believe that it is the case that language experience actually shapes this region. We ran two separate analyses. First, we associated language experience with the thickness of this region, and we saw this association. And then to the measure of language experience, we added information on the overlap between languages' phonological inventories. We told our model that language experience of these people has this form, and then we additionally controlled for the variability of sounds across their different languages. And it turned out how many languages someone knows and how different those languages are between each other is better linked to the thickness of the auditory cortex in these people. So the more languages someone speaks and more divergent those languages are, the thinner the cortex will be. And this is exactly why we think that it might be the experience shaping the auditory cortex, because it's more probable that the languages people speak were not chosen as a function of someone's predispositions, so someone's inherent brain structure. A lot of bilinguals, multilinguals in our study were brought up with different languages. So they didn't really have a choice as to which languages with which sounds to learn. So this is why we think that it's this varied, diverse language experience that actually shapes the auditory cortex.
Sounds like a very good argument to me. Olga Kopinska there. Sepsis is an overwhelming infection that triggers a dramatic inflammatory response that then becomes overexuberant and self-destructive. One sector of medicine where this is regrettably all too common is neonatology. Premature infants are particularly at risk. But what Ulla Beck and Ning Wen have discovered at the University of Copenhagen using pigs to model the problem is that blood glucose levels, driven by the sugar load in intravenous nutrition, seems to play a big role in sepsis risk. It's a fine balancing act though. There needs to be enough calories coming in to support growth and a healthy immune response, but push the sugar too high and it can spark sepsis.
We are very interested in the immune system of babies that have just been born, and particularly those that are born prematurely. Their immune system is not fully developed when they're born. So they have a tremendously high rate of infections just after birth. And these infections, they can develop into something we call sepsis, where the immune system overreacts and creates all sorts of problems. And that can be lethal to the baby. And at the same time, these very preterm babies, they also need some food, right? So often they will get what's called parenteral nutrition. which is nutrients that go directly into the bloodstream so that they can get the energy they need to grow. But this parenteral nutrition, it often contains a lot of sugar that increases the blood sugar levels. And we study how the blood sugar level and the immune response go together. Immune cells, to fight infections, have to use energy. So they can actually use the sugar that is provided in the parenteral nutrition to fight off the infection, which, if it's regulated well, is a good thing. But if the sugar levels become too high, then the immune cells react too violently. And that leads to this condition we call sepsis. So we wanted to figure out if we can lower the glucose levels in this parenteral nutrition in a way to prevent the development of sepsis.
It sounds like it's a necessary evil, Nin. You've got to have some sugar there to feed, grow, and fuel the immune response, but too much is a bad thing. How are you trying to study this?
We have developed a newborn preterm pig model, and we deliver pigs prematurely, and they will have a similar immature immune system compared to immature preterm infants. And we can deliver parental nutrition in similar manner as preterm infant receive. And we have produced a proof of concept study when we compare different regimen of parental nutrition with high and lower glucose than at the glucose level that preterm infants normally receive. We observe an increased survival of infected newborn preterm pigs receiving a lower level of glucose compared to the group receiving normal conventional level of glucose normal preterm babies receive.
But how low do you have to go to get suppression of the risk of sepsis, but you don't cause consequences of having low sugar? Because at the end of the day, if you become hypoglycemic, have very low sugar, that isn't good either.
So in this proof of concept study, we have tried to lower the dose 2 times lower than what preterm infant receive. And in this study, we have reached to a level of glucose into the bloodstream with the range of normal glycemia. So the at least preterm infected animals, they don't have hypoglycemia, whereas they still can maintain a good inflammatory response to infections and survive the infection well.
Ola, why did we have the level we did before then? Was that just because we'd always made parenteral nutrition to that sort of recipe, and because we'd always done it, we just carried on doing it? Why would we use something, a regimen, that put young babies at risk in this way?
Parenteral nutrition regimen is made because we need to give the infants nutrients to grow, right? But these guidelines don't take into account what happens when an infection enters the picture. Then the whole physiology of the baby shifts from growing to trying to fight the infection. So we actually believe that parenteral nutrition regimens should take this more into account, that the nutrition you receive while you are fighting off an infection should not necessarily be the same nutrition that you receive while you're growing.
And what sort of lead time do you have, Nin? Because taking into account what Ola's just said, that you need to grow, so having plenty of sugar on tap is a good thing when things are going well. But if an infection is going to set in and you're going to get sepsis, can you rein in the sugar after that process has already happened? Or is it trying to slam the door after the horse has bolted? Have you got to intervene before the sepsis kicks off and lower the sugar to get the beneficial effect here?
So this is actually a very tricky point. So in this study, we have actually tried to lower the glucose level in infected preterm pig after the infection symptoms show up, and actually it did not help to rescue the pig's mortality after infections there. And we think that it may be relevant for, for clinicians taking care of newborn infected babies, that they have to be aware of early symptoms of infections before lowering the glucose intake can be efficient.
And is that therefore the way to square this circle then, Ola? So you basically have to be very, very vigilant for any sign of infection, and then you would kick in with a lower sugar load in order to stop sepsis running away like this?
Yes, and you should also be vigilant about the glucose levels in at any time. And I think for clinicians, there's a great worry for hypoglycemia, too low blood sugar, because that has immediate acute consequences. Whereas hyperglycemia, when the blood sugar gets too high, is not an immediate consequence. Of course, we want to get it down into normal range, but it's not as an immediate problem if the blood sugar is too high. Whereas we should maybe think of it as an immediate problem. So it's a way of thinking of sugar as the fuel for the immune response. So you should try to keep it within the normal range.
Have you got enough evidence now to make this a sort of policy? Is this something that should become clinical practice off the back of what you're showing here?
It is already known that hyperglycemia should be avoided during infections. The new thing we provide here is that we can give some mechanistic explanation for why high blood sugars are associated with poorer infection outcomes. So that maybe gives a little more credence to the idea that hyperglycemia should be avoided. But it is already policy. But that being said, it is still a problem that occurs in the neonatal clinics all over the world at a regular basis, that blood sugar levels become too high.
A very important result, isn't it? Ola Beck and Ning Weng there. Finally, in this episode, can cancers control their hosts? Now, the reason for posing the question is that in many respects, tumours behave a bit like parasites, which have a long track record themselves of host manipulation. So, in theory, cancers may well do the same. In fact, in a previous episode of the eLife podcast, we heard how some tumours establish connections with the nervous system, leading to potential changes in host behaviour, perhaps accounting for the high rates of depression reported by cancer victims. Well, now another intriguing model has presented itself. A heritable tumour in Hydra. These are small carnivorous jellyfish-like animals which catch their prey using an array of tentacles. At the University of Montpellier, scientists have been studying a laboratory line of these jellyfish which, when they reproduce, which they do by budding off a clone of themselves, the offspring also inherit some tumour cells from their parent. These newborns show the unusual trait of growing double the normal number of tentacles, but not from the tumour cells. So clearly the tumour is somehow making the offspring do this. It's tantalising to speculate that the effect is to enhance the growth and reproductive potential of the host, which is what keeps the cancer alive. Animals without the extra tentacles don't fare as well, you see. The observations have intriguing insights into how tumours can manipulate remote host tissues, like blood vessels, to enhance their own growth. Justine Boutry.
We observed some jellyfish named Hydra that had different reactions when they had cancer. Some had cancer but had a regular number of tentacles, 7, 8 tentacles. Some had transmissible tumors that they inherited from a previous Hydra, from their mother, and they had much more tentacles, sometimes up to twice more, up to even 20 tentacles. We wanted to understand if it's the cancer by itself triggering these new tentacles to appear and what can be the benefit maybe for the cancer or maybe for the host if it's a reaction.
How do you know, though, that it's not a third option, which is whatever it was that caused the cancer to happen is also causing the extra tentacles to come along?
Indeed, there is also bacteria that can be present in some of the cancer, but in the transmissible cancer, we had 2 different lines, and one didn't have any bacteria inside their tentacles. So we grafted several types of tumors, some that triggers super or more tentacles in their original host that had bacteria, some that do not have any bacterial changes, and also some tumors that are completely spontaneous, not inherited, and that do not trigger these traits. And we grafted these very different types of tumors in new recipients to see how they will react, if they will develop or not more tentacles, and which one will develop more tentacles.
So you physically took cells that were the tumour cells from let's call them a donor animal, and put those into a recipient to see would just moving those cells across with or without any freeloading bacteria attached to them trigger the formation of extra tentacles?
Yeah, exactly. We took tiny pieces of the donor, we put them in new recipients, and we observed that only the tumors that were inherited in the past, that were already present in the mothers, with no correlation with any bacteria involved. Only this were able to trigger new tentacles in the new recipient, while the spontaneous tumors, they do not trigger the new tentacles to appear in the recipient.
And to be clear, the tentacles are growing from the recipient's own tissue. It's not that the cancer cells or tumour cells come in and they turn into tentacles. It's that they're sending some kind of signal that's making extra tentacles grow.
Yeah, indeed, they are sending a signal. We still don't know what is this signal, but at the end, it's still the recipient that is making new tentacles to grow. It's something that can be similar to what we see during metastatic processes. Sometimes tumors can trigger the formation of new blood vessels. It is still the blood vessel of the person that are growing, but it's the tumoral cells that are asking to the blood vessel to grow inside the tumor to brings more nutrients.
Is it that the tumour then is making the recipient make more tentacles because then it can catch more food and that's a bigger body to host a bigger cancer? Or is it that the organism is producing more tentacles, the recipient, in order to get more food so it can withstand having this freeloading tumour hanging off the side of it?
With more tentacles, they have more food and they produce more offspring. Because we have this correlation about producing offspring and transmitting the tumors to these offspring, it was very difficult at first time to understand to whom it is more beneficial. But what we've seen is that there is definitely benefits for both, but because it's the tumor that triggered this mechanism, it seems more crucial for the tumor than for the host himself.
There are some big animal transmissible tumours, only 2, dogs that transmit a venereal cancer through mating, and also Tasmanian devils that transmit a facial tumour through fighting. So might they be a useful model then to see if those tumours affect the behaviours of their hosts?
Indeed, we have these 2 models, and especially in Tasmanian devils, it's a very huge interest to understand how these transmissible tumors affect Tasmanian devil, because it's currently causing the populations to become close to extinction. So it's very timely to understand how they can manipulate Tasmanian devil even to be more transmissible, maybe making them more aggressive to each other, to be more transmitted. And there is also other kind of transmissible tumors that you didn't mention, which are happening in muscles and cockles. It's leukemia-like tumors, and they're also very interesting, and they can also help us to understand transmissible cancer more broadly.
Justine Boutry from the University of Montpellier. And that is where we have to leave it for this episode. You've been listening to the eLife podcast that's produced by The Naked Scientists. The previous editions, references and also the text transcripts for these programmes, as well as details on how, on the off chance you don't already, you can subscribe to this programme, are at nakedscientist.com/eLife. The Naked Scientists also publish a weekly science programme which covers the latest leading science stories, and you can find that one at nakedscientist.com/podcast. From me, Chris Smith, thank you for listening, and we'll hopefully see you soon in episode 100. Until then, goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.